All right, so I'm excited to talk to you all about Gillian Fiona Laundon, and to you all about Gillian Fiona Laundon, we'll get right into it. Okay. So Gillian was born in England, in Kettering, England, in May of 1938. And I don't know much about her early years, but in 1959, she completed a Bachelor of Sciences degree in botany from the University of Sheffield. So she went from Kettering, which is just north of London, a little bit north of Sheffield, and completed her bachelor's degree in botany. She then, with a bachelor's degree, went straight to the Kew Fungarium, which at the time was known as the Commonwealth Mycological Institute. And she worked there from 1959 to 1965. And it was apropos that you were already talking about Kew. Kew is probably the largest collection, although here the U.S. National Fungus Collections also has 1 million specimens and is the largest one in the Western Hemisphere. But Kew, I think, has more resources. So she was really prolific and jumped right into working at CMI. And one of the things that she wanted to focus on was rust fungi. And so she started to publish quite a bit on rust fungi. And so for those of you who are not super familiar with rust fungi, they are microscopic. They are microfungi, but they are some of the more charismatic and noticeable of the microfungi. On the left, you have mayapple and the mayapple rust, and this is a little later in the season when it produces teliospores, and we'll talk about the spore types in a second. On the right, you have mock strawberry, which are very small, and in the lower right-hand corner, you have part of a clothespin. But those orange pustules, those orange sort of cushions on the leaves are another spore type of a rust fungus. Those are Uridinea spores. And I think most people have seen the service berries with a rust on them, and those are actual ecial horns with ecia. So rust fungi have a very complex life cycle. They often have two hosts. They begin at 0 with spermatia, which join with receptive hyphae to grow within the plant. They produce a structure called an ecium with eciospores. That often goes to a different host plant. They produce a structure called a uridinium with uridinea spores, which can keep reinfecting that plant during the season. Later on in the season, they often produce a telium with teliospores. Those produce basidia with basidiospores, which then go to the initial host. So they're very complex, and they are some of the most, like I said, some of the most charismatic, the prettiest of the fungi that you can see under the microscope. Over here, we have eciospores in chains, uridinea spores, and teliospores, just as an example. So from 1963 to 1965, when Gillian was 23, 4, and 5, she wrote Rust Fungi 1, 2, and 3, which were mycological papers that monographed rust fungi on different host families. She redescribed the fungal genus Uridopeltis. She described a new genus, Angusia, and she wrote a number of descriptions of fungi. So this was one of her early publications from the Kew Gardens, the Rust Fungi on Acanthaceae. Came out in 1963. It was about 90 pages long. And she just started systematically going through that giant collection and looking at hosts, starting alphabetically with Acanthaceae, and taking out samples and looking at them to see what the rust fungi looked like, and writing descriptions and making drawings. And she was making the drawings with a camera lucida, which attaches to a microscope and projects what you're seeing on the slide onto the table so you can outline on a piece of paper what you're seeing. So all of the drawings are hers. She was a really great illustrator as well. And one thing that's also interesting is at the end of this paper, she talks about trying to quantitate what she was seeing, quantitate the length of the spores, the width of the spores, how many spikes they had. And she wrote a computer program to do numerical taxonomy and try to sort those quantitative features. And she says that these data were analyzed on the Elliott 401 electronic computer, which was in Rothamsted. And that computer was the very first computer that was used in public research. Only one, as far as I know, was ever made. And so she wrote a computer program to try to do the numerical taxonomy using the Elliott 401. These were two that she worked on while she was there. She redescribed Uretopeltis, which appears as these little brownish rust pustules on the underside of this leaf. And she described this new genus, Angusia. And that's what it looks like in more modern images. One thing she was also involved in was writing these descriptions of fungi and bacteria. And so CMI at the time, what is now Q, started putting out these descriptions. People couldn't always get access to journals. You couldn't always get access to decent illustrations. And they started putting out 10 at a time illustrations of fungi and bacteria. And I still use these today. So sometimes if I think I'm looking at something and I want to find a good description and illustration of the fungus that I think I might be looking at, I will go to a CMI description. I hadn't realized before I put this together that she actually was one of the co-authors on the first one ever, which was on coffee rust, Himalaya vastatrix. And so after that first one, she ended up writing 40 more with different co-authors. So after a really prolific time at Kew and working hard there, in 1965, she and her family emigrated to Laundon, New Zealand. And I'm not sure why they decided to go to New Zealand, but I was on the end of a phone call once where someone offered me a job in New Zealand and I said yes without hesitation. So she may have been in the same boat. Laundon was a small town then and is a small town now. It had a population in the 60s of about 8,000 people, has a population now of about 20,000. So it must have been a big difference from London. But it is a beautiful location right there at the base of those mountains. And she began to work at the Laundon Horticultural Research Center. At the time, it was one of the main horticultural research centers in New Zealand, probably the main horticultural research center. They also determined quarantine standards for imported plant material, which was what I was involved in when I was in New Zealand, and did diagnostics and identified diseases for people in the area, growers, and also homeowners. So I'd known about Gillian when I moved to New Zealand and when I lived there. And in years subsequent, I've had reason to go back to many of her papers, and I've gone back to some of her specimens as well. From 1967 to 1978, while she was in Laundon, she continued being very prolific. She published 74 new host records, so new hosts for different fungi. She published 27 new to New Zealand records recording for the first time certain fungi in New Zealand. She wrote 29 taxonomic notes that clarified morphological characters and species delimitation. She described 6 new species. She continued writing descriptions of fungi and bacteria. She wrote a chapter on rust fungi, on the Uridiniales, for the taxonomy of the fungi, which was a 5-volume treatise on fungi. I've got some pictures in the next couple slides. And she collected or identified over 600 specimens that are now in scientific collections. And you can go to archive.org and look up Gillian Laundon and find her collections. Going through her publications, I took the opportunity to sort of revisit a lot of her publications and see what was in them. And a few things stuck out to me. So this, again, was the 5-volume treatise on the fungi that was written in the early 70s. A number of my colleges got together and decided to write everything that they thought everyone knew about fungi. It started with volume 1, the fungal cell, volume 2, the fungal organism, volume 3, fungal populations, and then 4A and 4B were taxonomic treatises of really myxomycetes through basidiomycetes through mushrooms. So all the knowledge of the world's fungi were meant to fit into these 5 volumes. And she wrote the chapter on the Uridiniales. And it includes a key to all of the known genera at the time. And it also includes this table, which was based on a paper of hers in which she tried to make sense of some of the rust fungi that have unusual life cycles. Like I said, most of them make 5 spore types. They go to 2 different hosts. but some of them only make 3 spore types. Some do carry out their entire life on one host. There are a lot of variations. And so people for a long time were trying to come up with names for what is that life cycle? What is it if you only see 3 of the spore types? The one that she came up with is the one that's pretty much in use today. There was some argument between her and the Americans, Arthur and then Cummins, but I think she prevailed. One of the other records that I liked seeing was in a 1970 publication, she had the first record in New Zealand for a fungus called Mycocentrospora acerina. And that causes licorice spot of carrot and these black lesions on carrot that you can see here. Under the microscope, it makes these really nice spores, these conidia that are long and multiseptate and have these little appendages on the end. It's a really distinct fungus when you see it. And that collection, the first record, had come from Ohakune. And Ohakune is known as an area where they grow carrots, but it's also got one of the New Zealand icons, kind of like kiwi birds and kiwi fruit, and that is the giant Ohakune statue. So I thought it was just very fitting that that record had come from Ohakune. Another thing that she wrote about that I was interested in is in 1973, she wrote her first report of the rust fungus Commenciella mirabilissima on Mahonia, which is, I think, false holly. The earliest specimen had been collected in the 50s, but that had been in post-entry quarantine. And then they collected some out in the world, in the field, and they destroyed all those plants in the hope of eradicating the disease. But fungal diseases are incredibly hard to eradicate. And once you see something sporulating on a leaf, it is probably everywhere. And that is evidenced by the fact that in 2012, Jerry Cooper put an observation in iNaturalist of Commenciella mirabilissima in Christchurch in New Zealand. He had some really nice micrographs and some beautiful photographs of the symptoms. And I thought it was interesting. Jerry Cooper is a mycologist in New Zealand. He's excellent. You can see by the quality of his photographs, he really knows what he's doing. And his observation was seconded by John Plischke. So the mycological community is pretty small, and everyone's looking at everyone else's stuff, I think. So while she was alive, I think people recognized what a good mycologist Gillian was and how special she was. And she had 3 species named after her. Puccinia londonii is a rust fungus found on this plant, Thunbergia. It was found in Kenya in 1970, and the author, Leonard Holm, gave it the name Puccinia londonii. He said, "It is a pleasure for me to dedicate this new species to Dr. G.F. Laundon in recognition of significant contribution to uridaeneology." These are the teliospores of that rust fungus. Another expert in a group called hyphomycetes, which they make the spores without being inside any enclosed structure. They kind of look like fuzzy things on the substrate on whatever they're on. MBLS named Camposporium londonii after Gillian Laundon. And I only have this illustration of the one that was named after her, but Camposporium are saprobes, and they kind of, like I said, live like this. They might be overlooked. They're just these fuzzy things on bark, on logs, on stems. And Camposporium londonii was described from dead rose stems collected in New Zealand. The third fungus named after Gillian Laundon was Phoma londonii, and that was named in 2002. And this is an example of a phoma. These are leaf veins here, and this is the edge of a leaf. And if you mount this little black spot, which is about 50 microns in diameter, and put it under a microscope and squish it, you can see the fruiting body, the little hole where the spores come out, the ostiole, and then all the spores around. All phoma look pretty much alike. They're very hard to distinguish under the microscope. These 12 that are illustrated here, you can tell all pretty much look the same. So people are using DNA sequencing to try to separate species. And one thing I found interesting is there are a number of DNA sequences of Phoma londonii, but if you look at them amongst themselves, they are nearly identical. They show 100% identity amongst themselves. But the next highest number is 96%, which means that these are actually quite different from anything else that looks similar and maybe in their own group. And I thought that was pretty fitting for Gillian Laundon. You may also see a lichen genus Laundonia that is not named after G.F. Laundon, but her brother, lichenologist Jack Rodney Laundon. He was her older brother. And I think it's unusual to have both a lichenologist and a mycologist in the same family. And this has got to be one of the biggest sibling rival reflexes that you can have, that, you know, 3 species were named after me, but a genus was named after the other one. So Gillian moved to New Zealand, and in 1977, she submitted a piece of news to Taxon, the Journal of the International Association for Plant Taxonomy. And under that, they had, you know, awards and things about symposia. And she wrote in to say that Mr. G.F. Laundon of the Plant Health and Diagnostic Station Laundon, New Zealand, has changed sex as from the 22nd of January, 1977, and is now known as Miss Gillian Laundon. She continues her work in plant pathology, mycology, and nomenclature. And that may be the first and only time that someone has come out as transgender in a scientific journal. And I think it was incredibly brave of her to do so at any time, but especially at that time. But it also was just sort of, this is how things are. This is what it was. And she was very practical about it. I'd known about Gillian's scientific works, and I'd known that she was a transgender woman, and I wanted to know more about her. And last summer, when I was in New Zealand visiting, I had a chance to go to the Kawe Mahara Queer Archives of Aotearoa, formerly the Lesbian and Gay Archives of New Zealand. And they had a folder of a number of papers. I didn't know what I'd find. It was just a folder of her papers. It was actually under the name Gillian Cox, because socially she took her wife's name, but she kept the name G.F. Laundon, so that when she published papers from the time that she was Jeffrey Frank to the time that she was Gillian Fiona, the publications would look like they were authored by the same person. So it was a great collection, a lot of handwritten notes, a handwritten autobiography, a handwritten speech that she gave to her coworkers at Levin. It was very, it was just kind of amazing to see in her handwriting her thoughts on that part of her life. She had a portion of her handwritten autobiographical statement where she went in and took tea into her wife, sat down and said, how would it be if I changed sex? And that was her wife's introduction to Gillian and the beginning of a radical change in her life, possibly in anyone's life. She said she broke down and cried, and that at least her wife knew it wasn't a joke. In there were also an obituary written in a lesbian and gay journal of the time, and information on her organization transformation. So not only did she prolifically publish in mycology, but when she transitioned to a woman, she also created this organization with her wife, in which she prolifically published on all sorts of topics about being transgender. And she said, if you write to me at 5 Windsor Place, I will send you all the information that I have, everything I've learned, to try to make it easier for anybody else out there. Again, at the time in the 70s and 80s, there was not a lot of information. There weren't a lot of large communities. There was, of course, there was not the internet. And she wanted to get out there as much information as she could on what she had learned about the process. In there, she also talks about in 1975 coming out for the first time in public at a Christmas social at work. She attended dressed in a wig, high heels, and a dress that her wife had made her. She said her boss, with whom she had worked for 10 years, didn't recognize her, neither did most of the 100 others. There were a few unkind attempts to embarrass her, but without success. And this was from the talk that she gave to her coworkers, in which she said, it's not just a question of whether I'm going to be allowed to use the ladies' toilets, it's the attitude behind problems such as this, and the degree to which I'm going to be accepted or rejected by people I always thought of as my friends. So while her wife was completely accepting and her parents were very accepting and her children were very accepting, her coworkers were not always as accepting, and she apparently had some challenges to get them to accept her when she came back as Gillian. On February 8th of 1984, plagued by depression and debilitating headaches, Gillian ended up taking her own life, which I think was a tragic early end to an amazing life. But her legacy and work live on through her many scientific publications and through the continued work of trans people and mycologists in New Zealand and beyond.